Hi, thanks, Philippa. You kind of beat me to it there because I was going to say it's an honor to be here and a real treat to talk about science stuff in not kind of too serious academic environment, and that I am a mere microbe in the gut of Pierre Huyghe because I have researched and produced pheromones for some of his exhibits, and I'm not even sure if he knows about that or my existence. And I like the fact that these chemicals are traveling across the Atlantic and being put in the Palais de Tokyo or whatever, but coming across from Rockefeller. So I provided the squalene for his show at the Artists Institute in New York, and I wanted to draw your attention to this publication that was put out by the Artists Institute, which is a beautiful object by friends Alex Benenson and Jenny Jasky, and a lot of subjects and articles in this issue, I think, would be relevant to a lot of what is being spoken about today. And indeed, there's an excerpt in the program of this essay that I wrote called Self of Sense, where I wanted to question the notion that neurons are the only units of cognition or memory or learning. Why have we chosen to ascribe such importance to these cells and not other cells, such as the immune system or endothelial cells? And in doing so, I couldn't help but sort of knock up against the barriers or notice the blurred barriers both on the inside and the outside in terms of microbiome, the immune system, the interactions between the immune system and the nervous system, and then how this might be influencing our behaviors spoken about earlier. And also externally, how our sense of ownership or self can be manipulated, such as the rubber hand illusion, where you ascribe ownership to a fake arm. Even if you know it's fake, you will withdraw your arm in response to it being stabbed, or you'll perceive an ice cube even though it's not your real hand. And I think it's clear today that the boundaries of self are fluid, and I didn't want to keep ramming that home. I agreed to do this talk not knowing really who else was going to be here, and then I saw I was going to be last, and I thought, what am I going to add? I mean, I saw Daisy read from her book in New York in the summer. I thought it was like one of the best things I've ever read. People have written about this so much more eloquently than I could. So I thought, what can I really contribute to this symposium? And I'm just going to give quite an honest account of my experience as a scientist. And the point I want to make is maybe obvious. It's maybe so subtle that you will not even consider it a point. And this is an excerpt from a book that I recommended to the organizers, which is Ghost in the Machine by Arthur Koestler, and he's again, more eloquently than I, wrote basically my MIT essay, but unfortunately I came across it after I'd written it, because I was introducing this film called Ghost in the Shell. The original manga book was based on this book, and it's an excellent book. I'd recommend you visiting it. But I'm going to maybe state the obvious with some pretty pictures and some personal anecdotes. And throughout the talk, you might be thinking, well, what's the point? And just as a circle has no point, maybe don't even look for the point. Maybe just read my blurb in the booklet about what I want this talk to be about, and you'll maybe try and get what I'm trying to say. So the inspiration for the talk actually came from the title of the symposium, and I got this lovely email, and I didn't know, but the pufferfish make these pretty circles in the bed, because when I think of pufferfish, I think of TTX. So raise your hand if you know about TTX. Okay, that's okay. I'm really pleased that I can only see one hand. I don't know. So, do you know that pufferfish sushi is actually a very highly prized delicacy because the chef needs special training to prepare it, because the pufferfish contains this toxin. And it's deadly. It's completely, you need 1 microgram, I think, to die. Like you need TTX the size of a grain of salt will kill you. And TTX has been killing people for centuries. But I would say to a neuroscientist, and especially an electrophysiologist, TTX is the most magical, exciting compound, useful, and really, maybe this is too bold a thing to say, contributed massively to the view we have of the nervous system today, dare I say, the whole physiology of ourselves. And so this is the molecule. So these two chaps, just as I kind of like the emotional element that Phoebe was, her hero was Limagulus, and she is now in the same classroom, I'd say my heroes are Hodgkin and Huxley, and maybe learning about their experiments was as defining for me as like learning about the periodic table. And I did my PhD at UCL, and a lot of really key experiments were conducted at UCL in terms of elucidating the mechanisms of the electrical impulses that nerves conduct. And so this is a video showing some voltage clamp experiments. These were actually conducted in Plymouth. And this is showing that the membrane of a neuron can be depolarized when you inject current, and this leads to the flow of current through the membrane. And this was actually done on a squid axon, a squid giant axon. So our nerve fibers are tiny, but a squid giant axon is massive. It could, in diameter, can be up to like 1 millimeter, which is massive, big enough to get the electrode in, right? Which is what you just saw earlier. And so, again, it's comforting to say, oh, wow, the squids taught us so much about the nervous system. But I kind of, again, want to make the subtle point. I'm not talking about, oh, let's chop up other animals because we can't chop up ourselves. Or let's chop up other animals because their axons are bigger. It's more about the TTX and the sort of molecules that are produced. So basically, this is another recording from squid axon. This is done by Cole. And this was done from the nerves innervating muscle. And basically, D is when TTX is applied. So TTX blocks this nervous transmission. And TTX was then sort of basically sealed the deal physically in terms of proving that there was a voltage-sensitive sodium channel that allowed the flow of sodium ions, which contributed to the action potential. Hodgkin and Huxley subsequently then characterized the different conductance of the different ions, and there were ion channels found for those subsequent ions. But really, without TTX, I think, would we have ever got there? What else? Now we can make drugs. We can synthesize drugs based on the structure of TTX. But we really needed those years of evolution of another organism having a molecule designed to kill us, basically, in order to show us this receptor, in order to allow us to tune into something, to use Daisy's metaphor, that was already always there, but how else were we going to see it? And so these guys, I don't know why the lobster's there, I think maybe just because they didn't want to have the squid, and they won the Nobel Prize. And again, people, the emotional element of science was mentioned earlier. it's really, I can't help but feel that walking down the same staircases, walking down the same corridors, sitting in the same lecture theatre as these guys for 5 years of my life, they're somehow part of the building, and really, it makes me think, would I have even done my PhD on what I did it on if it wasn't for TTX? Would the idea we have about nervous transmission be what it is today? Because then this leads to other venoms and toxins produced by other animals, and they actually all act on channels. They might not be the voltage-gated sodium channel, but they tend to be voltage-gated channels. And so here is just a fraction of some of the channels that exist. You can tell that scientists are incredibly imaginative in their naming of these channels. There's just KB1, KB2, we go on. So that's potassium channels, sodium channels, inward rectifying channels. And these are just the voltage-gated guys. And I actually work on partly, one of my projects now is on a channel called TRPV1, and that's what responds to capsaicin, as well as heat and cold. So modern pharmacology is completely revolves around this concept of a channel. And when you're taking any drug, it's usually designed to act on a channel of some sort. And it just makes me think these compounds have so accelerated our understanding of that, but have also drawn our attention to that, right? So a bit like what Daisy was saying, if we're going to somehow conflate channels with sounds, it's like what other things are going on inside of us that we're not paying attention to because we're so focused on the channels. That's great. There's so much to do on the channels. There are a lot of channels, but there could be a whole other way of being, a whole other way of existing, communicating. And similarly, I don't want to be sort of elitist about it in terms of a fish is more important than a plant, because I feel, we're again, learning about how amazing plants are. And so it was early on as well. Langley was using nicotine and curare and looking at the action on muscle. And these compounds would either contract the muscle or inhibit the contraction of the muscle. And he got his receptor theory from the action of these compounds. So it's like, if it wasn't for these other organisms producing these other molecules that react with us, that kind of rubbing up against, would the view of ourselves be different? The model we have of ourselves physiologically has been entirely through the lens of these molecules produced by these other organisms. And I also want to shift now to sort of second phase in my life. I was at UCL, I was an electrophysiologist. I was surrounded in that culture, and then I moved to New York to join Jeff Friedman's lab, and it was a completely different culture. No one else in my lab is an electrophysiologist. They probably will glaze over if I mention TTX. They might not even know, no, they will know who Hodgkin and Huxley are. But in my lab, I'm talking about, amazing Rob McKinnon, figured out the structure of potassium channel. But it's a different culture, and so different organisms have been helpful. So I don't know, maybe more people know about GFP than they do about TTX. So GFP is a green fluorescent protein that was extracted from jellyfish, and we use this now to visualize the cells. We can snip out the bit that DNA that encodes for this protein, and then express it in cells of our choice under specific genetic promoters. And so we know whenever the cell expresses GFP is of a certain identity. So you can even talk about, having a fluid identity of the self as what we consider our bodies to be. Maybe, even cells are having their own identity crisis. So here it is up close. And you can see at the beginning, we probably went a bit crazy with this. It just expressed GFP wherever we could. But again, the experiments that I'm doing for my postdoc would not really be possible without GFP, because if you want to draw attention again to the fact that the experiments were done in certain brain regions because of morphology, you would be able to look down the microscope and the hippocampus, the cortex, the cerebellum look like a very beautiful circuit. They don't need, a few old school stainings will reveal the architecture of these structures. But the hypothalamus, the area that I work in now, is just like a tangled web of you can't discern that well different structures from each other. So by using the genetics in combination with this tool GFP, you can express GFP just in your cell of choice. And so this is what happens. And so I can record now from a cell expressing GFP, and I know the identity of that cell, whereas before it would just look like a black and white mass of cells that all looked exactly the same. And so from the GFP, the structure from the jellyfish, we've improved on that and tweaked that, and now these are the fluorophores, and these are basically the palette of the scientist. And we can co-express these in the same organism. So some cells can be some colors, some cells can be different colors, and we can also tag them to different proteins of interest, which I can explain later. And I'm sure a lot of you have seen the image of the rainbow mouse, which is where different fluorophores were expressed at random. So this isn't actually really saying much about the identity of the neurons, but this at least allows you to visualize them in a way that you wouldn't have been able to before. So at the similar time when Langley was chucking on nicotine and watching muscle contract, Paul Ehrlich was studying antibodies and antigens using toxins. Again, these molecules produced by other organisms that we collide with that reveal something about ourselves. And he was also obsessed with dyes and obsessed with being able to visualize the thing. So again, it's a visual importance, but it's about being able to notice something, to pay attention to something that is there. It's there, but it's about bringing it into existence. And so what happens is now we use antibodies tagged to the fluorophores that I mentioned, and these are our paints. So it's not like we need to express them through the germline. We just wash over an antibody of that's to a protein that we're interested in, and then we add a second antibody on top of that, and the second antibody has a fluorophore attached to it, and then we can visualize where the protein of interest is expressed, and that also tells us a lot about the identity of a cell. Again, another tool that has been incredibly crucial, which is sort of following along more the lines of the Hodgkin-Huxley thing, is that because the membrane of the neuron is voltage sensitive, if you depolarize it, it will fire an action potential that will be propagated and lead to neurotransmitter release, acting on postsynaptic neuron, more and more communication. But the kind of holy grail is to very precisely manipulate the activity of neurons, of known identity. Again, identity is important. So we take a channel that's sensitive to light that's expressed in algae, and similarly to the GFP, we know the gene that encodes for that, and then we can express that in neurons of choice, usually associated with the GFP actually, so that we can visually see whether this channelrhodopsin is, and then we can use light to activate the neurons. So in the Hodgkin-Huxley experiments, they actually used current, right? Whereas now you can just shine a light on and activate neurons and see how that manifests itself in the whole organism, behavior of the whole organism. So this is actually the laser. So this is channelrhodopsin expressed in muscle, and the blue light is activating the channelrhodopsin and making the muscle contract. So the hope is that channelrhodopsin is going to be used to treat many conditions where, say, we understand that the activity of certain neurons has stopped, and then we can introduce that. In my lab, we're actually working on another technology where the channel is tethered to a ferritin nanoparticle. So the way to activate the neuron is with a magnet, but the principle is similar. Again, another organism that's very much more in the atmosphere of the lab in New York are viruses. I didn't know anything about viruses when I did my PhD, and these are used as vectors to deliver genes of choice. So this is an adenovirus, and these are just sections of tissue that have been from my experiments. So even though this looks, very simple, these are actually dopaminergic axons in an area called the lateral hypothalamus with orexin neurons. But just to make this image, there's an adenovirus there that contains channelrhodopsin 2. The channelrhodopsin is also co-expressed with TD tomato. And then the green cells are GFP, and the GFP has been stained with an anti-GFP antibody, which is tethered to another fluorophore. And so actually in that image, okay, the tissue is a mouse section, but there's a virus, a jellyfish, an algae, a chicken because of the antibodies that are used, a rabbit, and maybe a donkey. So it's kind of, it still seems crazy and wonderful to me, but these are the tools of every day. This is what we cook with in the lab. This is just like what we paint with. And I kind of wanted to draw attention to that there's a whole world, there's a whole connection of animals that we've forced, but also this connection has illuminated, quite literally who we are. But I wanted to get back to this guy, and the talk is, about this guy, and I also wanted to give you maybe a little bit more of what you were expecting, and to sort of to join whatever ends I had up to make it circular, because the TTX that the pufferfish produces is actually not produced by the pufferfish at all. It's produced by a bacteria that lives in the pufferfish, and that wasn't found out until relatively recently. So I did give you maybe a little bit of what you wanted. And this is such a TTX producing bacteria, but I thought to try and convey how the model that we've made of ourselves, to remind ourselves of that. Oh, here is some bacteria producing TTX, and the ones that are green are producing TTX, so it's stained for TTX. So that's showing that you now know to look at that image, not just go, oh, that's a bit of green. That's actually a bit of jellyfish in there. Okay, and similarly, this is an axon, and those red dots are the voltage-gated sodium channels. So that's an antibody against the voltage-gated sodium channel with a fluorophore. So I don't know what animals were used to make up that image. And the ion channels are these pores in the membrane. They are these very selective holes that allow ions through. And so I wanted to end kind of just to flip the symposium on its head and call it the circle of a mind in the shape of a fish. Yeah, thank you.